the overall process of the electron transport chain, we see all of these electrons that are coming from other areas of glucose oxidation. We have electrons coming from glycolysis. We've got a few coming from the linking step. We've got some coming from the Krebs cycle, specifically FADH2. Remember, FADH2 is the only carrier that is coming from the Krebs cycle. So we have 10 electrons coming from 10 NADH and then 2 FADH2 carrying electrons to oxidative phosphorylation. We have 6 oxygen. We have 28 ADP and phosphates. And then via this electron transport chain, we end up phosphorylating those 28 ATP and ending up with the oxidized form of these carriers. So NAD plus and FAD. And then we have water being released as a byproduct here. Remember, water is released because this is a condensation reaction. When we add that third phosphate onto ADP, we synthesize a small amount of water as a result of this being a condensation reaction. Okay. Now, I really want to stress on the purpose of oxygen. Oxygen is the final electron acceptor. If there's no oxygen present, then the chain halts. There's nothing standing by to grab those electrons from the final complex and keep that chain moving so that we can acquire that hydrogen gradient. Alrighty? And so the overall reaction here at the bottom kind of reflects this, the phosphorylation of 28 ADP, and then we end up with the oxidized form of our carriers, as well as ATP and water. Okay, so here is an illustration of what we just described. And this is the inner mitochondrial membrane that we drew out. This is the outer membrane. This is the intermembrane space across which we're getting our proton gradient. And this is what's actually happening. So the complexes are inserted in this membrane, as we can see. Now I pointed out that you don't have to know the names of the complexes, but as you can see, they're numbered here. There's complex 1, there's complex 3, complex 4. And then the most important complex is ATP synthase, which is the final complex. We pass these electrons from their carrier molecules to the first complex. When we pass them from complex 1 to complex 2, we simultaneously pump hydrogen ions from the matrix into the intermembrane space. They're passed along and along, and then oxygen must be standing by in order to finally collect those electrons so that we can keep this chain moving and continue to create our gradient. And as a result of having such a strong gradient, now all of these hydrogen ions want to get back into the intermembrane space, and they go through ATP synthase, which is what actually synthesizes ATP. Okay. All right. So now we're going to kind of wrap up this sequence of events, and I'm going to look at a case study to kind of apply some of this knowledge and put this to the test. So this is the overall events of glucose oxidation. Oops. Okay, so this is a really nice summary image to kind of summarize everything that's happening. It's got the 4 phases involved, really 3 major phases and this very brief linking step. We've got glycolysis. And so this is kind of a good study table. This is the details that I want you to know. I don't want you to know the individual steps of glycolysis or the Krebs cycle. I really want you to know what the substrates are for each of these processes. So the substrate for glycolysis is glucose. The substrate for the linking step is pyruvate. For the Krebs cycle, it's acetyl-CoA. And for oxidative phosphorylation, it's the electrons, okay? What are the substrates for each of these stages? I also want you to know what's being yielded. So what are the products for each of these phases? So we yield 2 net ATP for glycolysis. We also yield 2 NADH carrying electrons. We yield 2 NADH carrying electrons for the linking step, and then 2 CO2 as a byproduct. Now remember, this is happening twice because we actually yield 2 pyruvate. We yield 6 NADH plus carrying electrons. We yield 2 FADH2 also carrying electrons. We yield 4 CO2 as a byproduct, and then 2 ATP from the Krebs cycle. All of these electrons are making their way to oxidative phosphorylation. So 10 electrons being shuttled by NADH, 2 electrons being shuttled by FAD, and then that undergoes oxidative phosphorylation. It yields us 28 ATP, and as a byproduct, we have water as a result of this reaction. And we need oxygen here as a substrate as well. So electrons and oxygen are the substrates, and the products are ATP. We get the oxidized form of our electrons and then some water. Okay. Now you will see some discrepancy in terms of the overall amount of ATP that is produced. Some textbooks will tell you it's 32. Some literature will say 38. So that there's some variation in the value, not only from person to person, but from state to state. So depending on what your physiologic state is, if you're pregnant, dehydrated, if you're older, you tend to be less efficient at synthesizing ATP. If you're younger, you tend to be a little bit more efficient at synthesizing ATP. So you'll see variation in the value from literature to literature, from person to person, and then from physiologic state to physiologic state. So that's why we have this 32 to 38. But your textbook uses 32. So you'll see me use that, and that's what I'll expect you to put on an exam, 32. But I still want you to know, for future reference, if you happen to go into this field or study this further, that you can see different values for the total amount of ATP synthesized per 1 molecule of glucose, okay? Let's see. So there's a question here, if 1 glucose requires 38 ATP, why do we only get 28 to 32 from ATP at the end, or are they not related? Okay, so that's kind of tying into that same point that I was making. So we typically see, in terms of what your textbook is using, the kind of lower value is 32 ATP overall. 28 of those are coming from oxidative phosphorylation, 2 from the Krebs cycle, and 2 from glycolysis. Remember, 2 net from glycolysis because we have to insert 2. So that gives us 32 overall. And that's kind of the lower end of the spectrum. But you can see other literature, you can see other, physiologic states or other textbooks that tell you 38. And of that 38, 34 would be coming from oxidative phosphorylation, 2 from the Krebs cycle, and 2 from glycolysis. So the discrepancy is really in how much the oxidative phosphorylation or the electron transport generate. Some books will tell you 28, some books will tell you 34, and then the remaining 4 are coming from the other 2 phases, okay? For our purposes and what your textbook uses is the value 28, okay? So 28 from oxidative phosphorylation, giving us a total value of 32 ATP. All right.